Hi, everyone. I would like to welcome you to Clario's webinar on blood pressure evaluation in clinical drug development. I am Borio D'Orpo. I'm a cardiologist and Chief Scientific Officer for Clario. On today's webinar, we will address and discuss FDA's second draft version of their guidance on how to evaluate blood pressure changes as part of clinical development. Dr. Kleiman will give a scientific background, so why at all are we doing this? Jeff Heilbronn will address key changes in the guidance that we issued a few weeks ago as compared to the May 2018 version, and he will also address blood pressure assessment modalities. Dr. Ruda will discuss study design considerations and also statistical analysis of obtained results. And then importantly, Dr. Kleiman will end up this with discussing labeling and labeling implications based on the results of a blood pressure evaluation that most drugs or all drugs intended for chronic use will be expected to undergo. I will monitor today's webinar, and please be aware that it will be recorded. The recording will be shared with you after the webinars eventually. So by that, I would like to leave the table to Dr. Kleiman, who is a clinical cardiologist with several decades as it is within cardiac safety with ERT and now with its successor organization, Clario. Dr. Kleiman is Clario's Chief Medical Officer. Bob, please go ahead.
Thanks, Boyer, and thanks, everyone. Next, please. So this is the updated FDA draft guidance just released this past February. As I'm sure most of you know, the original guidance came out a few years ago. This is still in draft form, but the FDA has taken some comments and their experience of the past few years and made some modifications. Next. Just in case anyone thinks this is something new, it really isn't. We've been talking to the FDA about this for at least 10 years. And in fact, in almost exactly 10 years ago, we had an FDA CSR think tank discussing the issue of drug-induced increases in blood pressure. And at that time, the FDA did not make any blanket requirements about intense blood pressure monitoring, and they certainly didn't ask for something akin to a thorough blood pressure trial. But we know that over the subsequent years, the FDA has asked for ambulatory blood pressure monitoring for certain drug classes that have a class effect of increasing blood pressure, or for some specific drugs where there were non-clinical or early clinical data suggesting blood pressure increases. We had the first draft guidance in 2018, discussed by the FDA at a public meeting in 2019. And then FDA published detailed recommendations in their experience. And I'm sorry, that's January 2020, not 2022. Next. The revised guidance opens with the following paragraph. This guidance is intended to advise sponsors on the pre-marketing assessment of a drug's effects on blood pressure. Elevated blood pressure is known to increase the risk of stroke, heart attack, and death. The effect of a drug on blood pressure is therefore an important consideration in risk assessment and product labeling. And that new sentence at the end is really the crux of this. The FDA is obviously convinced that even small drug-induced increases in blood pressure are important and therefore need to be discussed in labeling. Next. Now, why does the FDA think that a 1 or 2 or 3 or 4 millimeter increase in blood pressure due to a new medication might be deleterious? Well, it's always hard to talk about individuals, but we know that on a population basis, the epidemiologic data absolutely demonstrates that for every millimeter increase in blood pressure, the rate of cardiovascular events, including stroke, MI, and cardiovascular death increases. Next. Furthermore, we know that for a given blood pressure increase, the actual increase in the number of cardiovascular events is very much influenced by the underlying cardiovascular status of the patient population. This is a figure from the guidance, and it shows that for a very low risk subgroup, the equivalent of healthy young subjects, or patients with acne, for instance, even a 6 or 7 millimeter mercury increase in blood pressure is not going to really have a dramatic effect on cardiovascular events. In contrast, in the high risk subgroup, even a 2 to 4 millimeter increase in blood pressure is going to have a significant effect. So when we think about the significance of a blood pressure increase, we have to think both about the magnitude as well as the patient population being treated. Next. So why the concern? Well, the epidemiology is referenced in the guidance. The guidance also references the data from antihypertensive drugs studies, which have shown that treatment of high blood pressure reduces cardiovascular event rates. And we also know that there are several drugs that in very large studies were shown to have both blood pressure increases and an increase in cardiovascular events. Next. But to be perfectly candid, is there really any hard proof that small drug-induced increases in blood pressure are harmful? And the answer is not really. There have not been any well-controlled trials conclusively demonstrating that if there is a small increase in blood pressure due to the drug, that is what alters event rates. And it may even be impossible to do such a trial. But as the absence of proof of an effect is not proof of the absence of an effect, we still have to make decisions and we still have to act on the best available data. And I think that the FDA concerns really seem quite reasonable. And what's going to be critical will be the implementation and how these data are used. I'm going to stop there and go back to Dr. Darpo.
Thank you so much, Dr. Kleiman. And by that, I would like to hand over to Jeff Heilbronn. Jeff has a Master of Science in Health Promotional and Disease Management from the American University in Washington, D.C. Jeff has extensive experience from studying drugs, hemodynamic effect of drugs, and blood pressure evaluation. He has presented at several meetings and also co-authored papers on this topic. And Jeff will address the key changes of the revised second draft guidance as compared to the May 2018 guidance, and also then blood pressure assessment modalities. Jeff, please.
Thank you very much, Dr. Darpo. I appreciate the introduction and the opportunity to present the information today. So if we go on to the next slide, please. So again, as referenced, my goal today is to provide you with a look at both the 2018 draft guidance and the recently released 2022 update, as well as the modalities. So this slide just provides, again, kind of a quick summary that Dr. Kleiman had shared. One of the things that I did want to highlight here in the publications is that following that 2019 open public discussion on the original draft guidance on the 2018, Dr. Garnett and colleagues at the FDA did publish a very valuable article looking at redefining the blood pressure assessment and the role of ABPM for drug safety. This article provides for some great case studies and examples of looking at that pressor effect, that blood pressure increase in specific drugs. And then again, as we're looking at today, we'll take a look at the updates that have come out with that February 2022 draft guidance. So if we go on to the next slide, please. So looking at the 2018, the initial pressor effect guidance, it did point to a couple of key considerations. And for many people, this was new, although it had been under discussion going back, as Dr. Kleiman had shared, to 2012 and beyond. The focus here was an increase in blood pressure. This was not the fact that a compound created a hypertensive state, but the fact that the goal would be that there was an off-target effect that would relate to an increase in blood pressure. And this increase in blood pressure is an important consideration to the benefit risk assessment of the drug in development. What was also important to note was that there was a differentiation in the therapeutic treatment. Was the intended therapeutic agent intended for a short-term prescription or allocation where the individual would be on it for a shorter duration, or was this a therapeutic treatment that was for more long-term or chronic exposure? So taking a look at something that might be given for a lifetime, such as a ADHD or other neuroscience, and taking a look at that increase in blood pressure. Specifically in the 2018, it pointed out the systolic blood pressure as the primary blood pressure value of interest. The other bullet point that you'll see there is that introduction and the reference to ABPM, ambulatory blood pressure monitoring, being the primary modality for assessing that change, and the reasons behind that being associated with the precision and the reproducibility. Lots of lessons learned from drug development in the hypertension world and applying them now over to the safety assessment for that pressor effect, and again focused on an increase. There's also reference in the 2018 to the value of the clinic BP assessment and taking a look at triplicate assessments in an office setting. One of the things that was important to highlight in the 2018 was that it was clear from the regulator's perspective that the method for assessing blood pressure were not as consistent and not always as adequate. So really a highlight on the modalities and methodology of taking blood pressures. One of the things that was also important to note, and Dr. Kleiman made reference to this, and it is important then when looking at the draft guidance that the intent is to look at the intended study population, the therapeutic population and or similar population. And when necessary, to then take a closer look at the subset, which became even more clear in the 2022 release. The idea being that if you had somebody on a rheumatology therapeutic compound and there was an increase in blood pressure that was observed, well, it's very likely that some of these participants may in fact be diabetic or may be hypertensive, and that it would be important to take a look at a subset of the population that were at a higher risk. One of the things noted that was missing from the 2018 was actually a therapeutic threshold or a regulatory threshold, which again is addressed in the updated release in 2022. So let's go on to that next slide and we'll take a look at the 2022 and some of the things that were unique or updated in that release. One of the things to note is that in the 2022, we do see a reference to diastolic blood pressure values. However, throughout the evaluation or analyses, there's still a primary focus on the systolic, but it does introduce the diastolic being an important component as well. There is emphasis now on the clinic assessment, again, and the value of taking a look at a potential dose or exposure response relationship at a clinic level, as well as identifying a larger increase in a short-term exposure to the therapeutic agent under development. There's a lot to go into that on the clinic BP assessment as to the approaches and standardization of that process, which we can follow up at a later date. Going down to the next bullet point that Dr. Rudo will spend some time on is taking a look at the clarification on the study design and going deeper into the discussion around whether an active or a placebo control group needs to be included in this. And then on to the statistical analysis. And clearly here was one of the areas that the FDA had identified that was lacking, where now there's identification in the updated draft guidance that we're looking to exclude a 3 millimeter increase in the 24-hour average systolic pressure. In addition to that specific information, there's also a highlight in looking at the 24-hour systolic mean, as well as now including the daytime and nighttime average values in the analysis plan. As both Dr. Darpo and Dr. Kleiman referenced, the labeling implications is probably the most interesting addition to the February 2022 draft guidance. Lots of references as to where and how to represent that increase in blood pressure if in fact it is seen during your assessment, and Dr. Kleiman will go into more depth on that. So if we go to the next slide, please. So with that, for some of you, this may be a bit of a review or elementary. For others, it's going to be an introduction. But as we go on to the next slide, please, what I wanted to do is take the opportunity to introduce and give you an image of what these modalities are that are referenced in the draft guidance, but also to highlight some future areas of looking at blood pressure assessment and defining an increase or pressor effect. Looking at this screen, going from left to right, you can see that there's the clinic monitor, and I reference that as a static time point. So you have the scheduled visits, again, the reference in the guidance to take the triplicate readings. Very important when we look at this is that there needs to be standardization within a patient and across all study participants because of a means to reduce variability and ensure consistency in that assessment, especially if we're looking to define a potential large blood pressure change. The middle slide or image that you can see in this slide is referencing the ABPM, the primary methodology that is going to be proposed in the draft guidance and implemented into the study. A very powerful tool, lots of lessons learned from the world of hypertension. The strength of this being it added into the number of readings that can be captured over that 24-hour period, enhancing precision and reducing variability. Again, the benefit is that we can see both day and nighttime blood pressure response. The last slide, although not referenced in the guidance, is something that I thought was important, which takes a look at home blood pressure monitoring and the ability to capture data over a longitudinal period beyond just a 24-hour, and to say what could be seen over extended exposure and the blood pressure response. So going on to the next slide, please. I did want to share with you some of these images. What is it that we're actually measuring when we're implementing these technologies, and with ABPM being a primary focus of the draft guidances? This again is just an example looking at an oncology agent, not specific so much to the guidance, but was clearly showing a definition of what you can see with the ABPM data. Looking at the graphic display on the slide, you can see that there's a purple line being the baseline, and then 2 treatment days. So you can see that there's the systolic represented on the top series of slide, and then the bottom being the diastolic, and on the x-axis, the time of day being the 24-hour period, and the blood pressure on the y-axis. And so you can see that there is a change from that baseline, that non-treatment reading. But what was interesting to see here is that when you take a look at the 2 treatment days, that in fact there was no increase in the blood pressure response. This therapeutic agent and that class of agents is known to create an increase in blood pressure. And in fact, this using of the ABPM technology was a great way to define that change in blood pressure and representation over both day and nighttime. As we go to the next slide, this is a very simplistic look at the data, but this is the clinic blood pressure. And there's been a lot of discussion on clinic blood pressure generated even from the SPRINT trial, which was somewhat of a landmark trial when it came to clinic BP assessment. But the idea being, and the emphasis on the standardization in the process and the implementation of triplicate readings. But in addition to that, ensuring that there is a standardized rest period prior to the inflations, that there is time between each of the readings that is allocated. And again, one of the things that is still open within the draft guidance, both in the 2018 and the 2022, there is no specific reference to whether or not it has to be used with an oscillometric or automated device, or whether auscultatory using stethoscope and Korotkov sounds is the way to go. I would say from our experience, we've seen more of the oscillometric and standardized process because it does reduce some variability and some bias that can be seen with an auscultatory assessment. But there are systems out there that do allow for that standardized process as well as electronic capture of the office BP data. If we go on to the next slide. This was, again, just an opportunity to introduce another modality, this being the telemonitored home BP. As you can see here, looking on that x-axis, you can see that the duration isn't in hours anymore, but you're actually looking in days to months. So again, depending upon what the blood pressure response is, there are many ways to define that signal. This happens to, again, to be able to provide that extended exposure and has been used in clinical trials and is complementary to the other modalities that we've discussed, but something to keep an eye on, again, not in the guidance, but a valuable tool for looking at blood pressure changes. And then to the last slide, thanks. This was just a chance to put them side by side when looking at the ABPM, which again is the primary modality outlined in the draft guidance, which would be then supported by the clinic. You can see the value of this in this very simple display. Each of those lines on that left-hand side graph shows you the top of the line being the systolic, the bottom being the diastolic, and each of the dots representing the heart rate. In this, you can see that we can really establish a profile of a blood pressure change from baseline, even capturing that day to night and that circadian change in blood pressure, which can be important, especially when we start to look at defining the blood pressure in a specific subset of a therapeutic population. Over on the right-hand side, again, is that image of the home BP, again, giving the opportunity to both get that immediate 24-hour using the ABPM and defining that blood pressure response as outlined in the guidance, but also further down the road being able to take a look at an extended definition of a potential blood pressure response. So with that, I'd like to thank you and hand the presentation back over to Dr. Darpo.
Thank you, Jeff. Thank you so much. And by that, Dr. Rudo will address design considerations and statistical analysis of obtained data. Dr. Rudo is a cardiac electrophysiologist with more than 15 years of experience from both clinical practice and pharmaceutical industry. He serves as Clarios Vice President of Cardiology and Deputy Chief Medical Officer, and he works with sponsors and CROs, of course, on cardiac safety issues in drug development. He also oversees our statistical group and medical writing group. Dr. Rudo, please.
Great. Thanks, Dr. Darpo. And thanks, Jeff, for kind of teeing things up nicely for me there. You can go to the next slide, please. So you've heard now about the sort of 3 different tools that we have for blood pressure assessment in clinical trials. 24-hour ambulatory blood pressure monitoring, home blood pressure, and clinic blood pressure. And Jeff's also shared, what the data can look like from these different modalities. So what I'd like to do is spend a little more time just describing the use case for each and how they can be applied across the different phases of development. As was already mentioned, there's some new language in the guidance that's particularly relevant on this topic, and I'll highlight that as we go through. So starting off with 24-hour ambulatory blood pressure monitoring, we've heard how this modality works, essentially collecting near continuous data every 20 or 30 minutes over 24 hours and providing a very comprehensive set of information rather than relying on individual time points. This remains the gold standard for regulatory submission purposes. As mentioned, there's a role for clinic blood pressure now, also from a regulatory perspective that's described, but generally this is still going to be the gold standard, particularly in assessment of drugs for chronic use. And that's now, quite clear and re-emphasized in this version of the guidance. Why? Well, ABPM offers several unique advantages. So first of all, the assessment over 24 hours provides insights to both daytime and nocturnal blood pressure. The higher volume of data gives you more precision or ability to detect small changes, which, as we've now heard, a change as little as 3 millimeters of mercury can become relevant from a risk as well as a labeling perspective. Custom schedules of inflation sequences can be set, so you could have a higher frequency of inflation during the daytime versus nighttime if desired, based on the PK of the drug. You could make adjustments as well. And importantly, with this and the other automated modalities, is freedom from bias that's often introduced by human nature to sort of round up or round down, depending on the use case. So several advantages to 24-hour ABPM as a modality. The guidance does set some specific recommendations for inflation sequence with a minimum of twice per hour over 24 hours. There's also mention that the sessions themselves should be scheduled appropriately based on the PK of the drug and based on the steady state and when the maximum effect on blood pressure would be expected. So there's some specific language that gives guidance in terms of how to apply this best, within your study. Can go to the next. Home blood pressure is a little bit tricky, as there's really no clear consensus in how to best utilize or incorporate this type of data into your development program. There's certainly several key advantages that we can discuss, though. Most obvious is that you can, compared to ABPM, you can get blood pressure data at home, but over a much longer period of time with far more time points. So less readings per day, obviously, but many more days of data. And you can do so without increasing the burden to the patient participating in the study by increasing the number of site visits to collect that data. So with that context, most would agree that this is a relevant technology when thinking about decentralized clinical trials or hybrid clinical studies that have some mix of home visits as well as in-office visits. This also lends itself, particularly well to kind of early mid-size phase 2 studies, perhaps, as well as supplementing time points in larger phase 3 studies, which have limited clinic visits, again, with the ability to sort of fill in those gaps between visits, but still have insight as to the blood pressure data. Another key advantage is the concept of these being real-world measurements, as opposed to in-clinic blood pressure assessments. So I think people are well aware of the potential impact of catecholamines on blood pressure results. It's the reason that there's a standard 5-minute waiting period recommended before any blood pressure assessment, be that at home or in the clinic. But when you measure blood pressure at a clinic visit, that is not a real world setting. So the patient had to drive to the site that day. They probably have angst over some other tests that may be scheduled that day for them. They may be receiving some results that day, either nervous about receiving the results or upset about results they just received, versus assessing blood pressure on a sort of normal day that they would have. So home blood pressure data certainly has advantages over clinic data from an accuracy perspective. And then lastly, when thinking about home blood pressure is the modality selection itself. So this could theoretically be accomplished just by having the patient record their own blood pressure on self-owned equipment and recording that in the log, by hand and bringing it into their next visit. But there's technology that can improve upon that quite significantly. So number one, using an automated device which has a pairing, if you will, with a smartphone that gives some instructions to the patient. So perhaps reminders on when the blood pressure assessments are expected, or reminders on best practice, that 5-minute waiting period I mentioned before. Cuffs that are used for this purpose within a clinical trial can be programmed for triplicate inflations rather than single readings, like an over-the-counter product would use. And very importantly, the data can be captured digitally and then transmitted automatically via Bluetooth or other technology directly into the clinical trial database. So the site would have, number one, visibility real time to these values, but also number two, there's no transcription errors to worry about in terms of manually collected data. So the right choice of technology when thinking about home blood pressure is probably just as important as anything else. We'll go to the next. Now the story with clinic blood pressure is starting to take some real shape, and as Jeff mentioned is emphasized in the 2022 revision to the guidance in more detail. So first, I think it's important to emphasize how this differs from just routine clinic vitals. Clinic vital signs, of course, are a part of any clinical trial, and how this is different is really the methodology in collecting that blood pressure data. So first, triplicate collections. It's now specifically mentioned in the guidance the value of triplicate versus single blood pressure assessments for these time points. The protocol should typically include 3 readings that are 1 to 3 minutes apart. And that should follow a 5-minute rest period, as I also mentioned, and the same arm should be used at each visit. Again, all sort of details that are now specifically noted in the guidance that were not present previously. Regarding use case, there's 3 examples that are provided. One is for the assessment of drugs intended for short-term use. So, that ABPM is definitely the modality that's appropriate or necessary for detection of a small blood pressure change for chronic use drugs, but is not required in the setting of short-term use drugs. And this modality could instead be used for detection, specifically detection of any large effect on blood pressure. And that's the second use case that's mentioned is as a general safety measure for assessing for any large increases. Properly performed clinic blood pressure can suffice. And then the last example is characterization of dose response. So, in a dose escalation study or in a phase 2 with multiple dosing arms in parallel, the role of this technology is critical in terms of detecting any difference in blood pressure effect across the different treatment arms. From a design perspective as well, the guidance states that the time points for vital sign collection need to be thoroughly considered. So Tmax, some drugs are going to affect blood pressure based actually in correlation with the PK profile of that drug. Some will have more of a long-term effect that doesn't actually vary with PK. But to detect that, you would need the right time points. So vital sign time point collection is critical, including covering Tmax, covering steady state trough, covering several time points over the course of the treatment itself. It's also mentioned, sort of analogous to ECG, not to use a screening blood pressure as the pre-dose baseline, but actually a specific pre-dose time point should be specified to serve as the pre-dose value against which all on-treatment data will be compared. So again, key advantages here for clinic blood pressure over routine vitals just comes down to reducing variability, improving the precision of the data, using a single model device across all clinical trial sites, setting a standard inflation sequence that all sites are using, capturing that data digitally, rather than relying on transcription manually into the clinical trial database, eliminating investigator bias, which I mentioned before in terms of rounding. These are all sort of advantages of using a formal modality as opposed to just relying on sort of more classic routine vitals, at a site. Can go to the next. So before moving on, just a few final comments on best practice for blood pressure collection, which is really applicable for clinic or home assessments. Again, since we're talking about detecting very small changes in a data type that inherently is actually quite variable or noisy, data quality is key. And so, how can we ensure that the best quality data is collected, reducing the risk, therefore, for any false positive or false negative safety signals to the development program? So first, multiple cuff sizes will be needed to ensure the right fit for patient, depending on your population, whether it's a pediatric population or a particularly obese population. That cuff size range may need to be a serious consideration. Patient positioning is important, using a device that prompts the patient or the site staff to make sure that the position is correct, as well as that waiting period is adhered to prior to that first inflation. Setting the inflation sequence to be uniform across both home and clinic visits if both modalities are used. Automation we've talked about in terms of the advantages of that to reducing bias. And then device selection, very important that the device that is selected for your clinical trial meets, the appropriate validation standards, that the software that is being used for analysis meets the CFR Part 11, compliance standards in terms of data protection and data privacy. Go to the next. Thanks. So, now that we've talked about the best tools for collecting blood pressure, best practices for using those tools from a clinical trial design standpoint, I'll focus a bit on how to analyze that data. You can go to the next. So this slide here just really lists out all the typical elements of a statistical analysis plan for blood pressure data. Systolic, diastolic, and diastolic blood pressure and heart rate are sort of obvious, and we'll talk a little bit more about those in a moment. The role for mean arterial pressure and pulse pressure is far less clear. Often part of a statistical analysis plan, often included in the data set from a centralized blood pressure tool, but perhaps not critical, certainly not mentioned in the guidance as specific elements that need to be assessed. Categorical outliers, however, are important for assessment. We'll talk about those in a bit, as well as Dipper status. Can go to the next. So just a word first about data cleaning. Despite best efforts and things that I just was talking about, there will be issues encountered in terms of data quality. Not only does blood pressure vary from beat to beat, but it's very sensitive to motion or activity when thinking about the collection modality itself. So for ABPM in particular, which unlike office or clinic BP where you have that rest period I mentioned, ABPM is set to trigger every 20 or 30 minutes, sort of regardless of what the patient activity level is at that time. And so it's expected that there will be some readings that are spurious or inaccurate due to patient movement, just as one variable really. That could be happening at the time of inflation. But you don't want to include those spurious values in your analysis, and also don't want to delete true data that just appears to be an outlier. So this is sort of a tricky topic. And I think the key here is recognizing that there are certain implausible values or implausible ranges, and pre-specifying those in your statistical analysis plan as data that will be excluded. So for example, if you have a diastolic blood pressure less than 40, or a heart rate less than 30, systolic over 240. Those are fairly implausible values, depending on, of course, the population, and these ranges will be adjusted for the population of your study. But you may need to consider this in terms of just ensuring that the data is properly cleaned, before finalizing your analysis data set. Go to the next. So primary endpoint in almost all cases will be a weighted mean average of change in 24-hour systolic blood pressure. That weighting is important because of, as mentioned, some missing inflations, missing values. You can have, potentially less values at night if the inflation sequence was set up to have, higher density of inflations during daytime hours. So weighted mean average is typically what you'll see in terms of primary endpoint. Tmax is mentioned here on the screen only because there will be a small subset of drugs where the blood pressure effect is immediate. It's actually directly correlated to the PK profile of the drug. And if that's the case, then your analysis may need to be skewed somewhat, not just to look at overall, but also to look at Tmax related changes as primary. Go to the next. So secondary endpoints, as Jeff mentioned, diastolic blood pressure has now become relevant in the latest version of the guidance, and should be that 24-hour weighted mean change in diastolic blood pressure is a key secondary endpoint. It's also mentioned in the guidance to assess day and nighttime effects. So those hours can be predefined in your SAP, and then that data analyzed separately as a secondary endpoint. Hourly change, categorical changes, which we'll talk about shortly, and also mentioned here is change in background treatment. So if a new blood pressure drug is started or a change in existing blood pressure therapy is made, that needs to be captured because it obviously will affect the data, and so that needs to be part of the analysis as well. Go to the next. So categorical analysis will vary, or at least these values, the thresholds will vary based on the treatment population that's being studied. But the concept applies, regardless of population, and that's to assess for an absolute change or change from baseline in a categorical way with respect to systolic and diastolic pressure. There's also a concept of dipper status, which can be assessed. So all of us during the overnight hours should have a normal physiologic decrease in our blood pressure of about 10 to 20% for systolic blood pressure. And in a categorical type approach, you can analyze dipper status, looking for so-called non-dippers or even reverse dippers to assess for a potential adverse effect of a drug beyond just change in over 24-hour systolic blood pressure. Go to the next. So, how do we contextualize all of this? Well, we're looking to learn really the magnitude of effect on blood pressure that a drug may have, the duration of that effect. The concept of subgroups is now introduced in the guidance. Looking for any subgroups that are particularly at risk or that a more notable effect was seen. Although perhaps not powered to do so, it may be apparent nonetheless. And then of course, how is the risk that's detected relevant to the specific patient population? And that's something that is unique to each drug, and that in determining that threshold of concern, therefore, will be unique as well. In terms of mitigation, so let's assume we do detect a blood pressure risk. What's next? Well, it depends where you are in development, really. That could affect next phases of development in terms of intensifying, use of vital sign or other blood pressure data in the next study. It could influence phase 3 study design, and then of course, from a labeling perspective or post-marketing surveillance perspective, there could be implications as well. Go to the next. Sample size. So the objective is to be able to detect a change in blood pressure that is relevant to that population. And the threshold of 3 millimeters of mercury is what is described, but there's also some language that says that may be different depending on the population under study. The desire specifically is not to detect or show the absence of a mean effect of 3 millimeters, but actually to exclude that the upper bound of the two-sided 95% confidence interval is beyond 3 millimeters of mercury. And so in doing so, we can calculate based on certain assumptions a sample size that would be necessary to detect that effect. And if we assume a 10 millimeter standard deviation for a change in systolic blood pressure, in an open label design, that would translate to 119 subjects in order to detect that 3 millimeter change. In a parallel study that has placebo, that sample size increases significantly up to 234 per group. And perhaps if time permits in the Q&A session, we could discuss this a little bit further. But certainly a challenge in terms of the size of these studies. And it's, again, comes down to the nature of this data being quite variable and therefore needing a fairly substantial sample size in order to detect small changes within such noisy data. So hopefully that was useful. And again, I'd be happy to answer questions if you enter them into the Q&A for us during the Q&A session at the end. I'll hand back over here to Dr. Kleinman now, who's going to take us through some really entirely new content in the guidance regarding labeling considerations.
Thanks, Todd. So now we're up to the end game, the label. Historically, the FDA has not emphasized labeling very much, really wasn't mentioned much in the initial draft guidance from 2018. At a public meeting in 2019, the FDA clarified a few things. They said, first, these data generally aren't going to be used for the approval decision unless your blood pressure increases enormous, but they'd be used to inform the prescribing physician what to watch for, how often to monitor, just to make sure that they're sensitive to this concern. They also said that a cardiovascular outcome trial wouldn't be necessary just because a drug showed a small increase in blood pressure. They were thinking that we already know that blood pressure increases are bad. It doesn't need to be shown again and again and again. And overall, over the past few years, the labels have generally been very reasonable. Next. So as an example, here's the Levantinib label from 2015. And in the highlights of prescribing under warnings and precautions, it simply says hypertension, control blood pressure prior to treatment and monitor during treatment. Withhold for grade 3 hypertension despite optimal antihypertensive therapy, discontinue for grade 4 hypertension. I think that's pretty obvious recommendation that people would follow this behavior anyway. Next. In the full prescribing information in section 5, there's a bit more text. There's some detail about the effect size, how these data were collected, but really very little prescriptive advice. Next. Here's the asciminib label from 2021. In the highlights of prescribing, hypertension, monitor blood pressure, and manage hypertension as clinically indicated. Interrupt, dose reduce, or stop if hypertension is not medically controlled. Eminently practical advice. Next. And when we get to the full prescribing information, again, details about the magnitude of the increase and where the data came from, but not a lot of firm recommendations. Next. But then we get to Xiasted. The label was revised in 2018. This is a testosterone supplement, and there's now a black box warning. And the black box warning states that this can cause blood pressure to increase with significant deleterious adverse events and death. One should exercise judgment before starting a patient on these supplements. Periodically, you should monitor for new or blood pressure, new hypertension, or new significant increases and treat appropriately. So this is much more firm than in the previous labels that I've shown. Next. And in the full prescribing information, there is usual information about the magnitude of the effects and some firmer recommendations about monitoring for blood pressure. So again, one could discuss whether this was appropriate or not. If you read this, you will notice that the mean effect was only a 4 millimeter of mercury blood pressure increase, not a tremendous increase. But given the underlying population, that would still lead to a significant increase in cardiovascular events. Next. So wording's taken straight from the guidance. There are recommendations about what should be included in the pharmacodynamics section. And the guidance goes on to say that a brief description of the ABPM study design and study population should be included regardless of whether the drug was shown to increase blood pressure. In other words, even if you don't have a blood pressure increase, the pharmacodynamics section should explain where those data came from. If you do show drug-induced blood pressure increase, then you would have information about effects on systolic and diastolic blood pressure with the doses studied, the distribution of blood pressure and increased sizes, if there's a dose or exposure response, and if you could detect differences in how various subgroups were affected. Next. And finally, it closes with, if the drug is associated with a clinically significant increase in blood pressure, see section 5C, then the following should be included in the warnings and precautions section. A description of the blood pressure increases, the clinical implications, and steps to take to prevent, mitigate, monitor, or manage blood pressure increases. I will point out that if you read section 5C, it doesn't really define what would be considered a clinically significant increase in blood pressure. It doesn't do that, I think, because the FDA would like sponsors and review divisions to look at the underlying cardiovascular risk of the target population before concluding whether or not a small increase is really clinically meaningful. Because as an example, for a population of elderly men, many of whom have hypertension, diabetes, maybe metabolic syndrome, may have pre-existing coronary disease, the risk enabled by even a 2 or 3 millimeter mercury systolic blood pressure increase may be quite significant. So that's all that we have for the prepared presentation. Oh, I'm sorry, I do have one more slide. Next, please. Got ahead of myself. The implications of the labeling suggestions are fairly apparent. The label should describe how you collected the data, the magnitude of the effects, adverse events, monitoring and mitigation strategy. And overall, this is a significant change from previous labeling recommendations. And in some ways, this is reminiscent of the type of labeling that drugs now have for QT effects. For good or for bad, I think it's important that we recognize this is here now. We have to collect this data, present it at the time of submission, and be prepared to put the relevant information into labels. So now I'll say that's the end of our prepared presentation, and back to Dr. Darpo.
Thank you so much, Bob. We have received a number of questions, and the first one I would like Dr. Kleiman to address is this real? I mean, how realistic is it to expect that this guidance will turn into final guidance, given that FDA has been discussing this for like 10 years? The first draft was in 2018, so almost 5 years ago, and now we have the second version. How real is this?
It's a very interesting question, and obviously, reading, as Yogi Berra said, predictions are dangerous, especially when they're about the future. Certainly that is true for predictions about what the FDA will do. And I don't know if they will finalize this, but what we know is this is a reflection of their current thinking and their current practices. So yes, it is real. You can expect more and more the FDA to start asking to see this type of very high quality blood pressure data for all new drugs that are given chronically. And it will be much more likely and to be more intense for drugs that target a higher risk population.
Thank you, Bob. Another question for you, actually. Is this all about small molecules, or are we also looking at large therapeutic proteins?
This is about any drug that has systemic bioavailability. There are obviously biologics that do have blood pressure effects, some increase, some decrease. So again, if it is going to be used chronically, then this type of data will be appropriate. If it's just for a single use and the pharmacodynamic effects don't last particularly long, then no, I think that kind of compound would probably be excluded.
Thank you, Bob. Dr. Rudo, could you elaborate a bit on what is a weighted mean? And also, what does it mean to exclude a 3 millimeters effect? What is it we're comparing? Is it baseline to drug, or what is it?
It's actually it's an interesting question and a challenging question, because it depends on the design of the study and what you're actually able to exclude or state. But what we're looking to exclude is a 3 millimeter increase in mean 24-hour blood pressure using an upper bound of the two-sided 95% confidence interval, right? So weighted mean, to take that question first, essentially you will have some hours during the day where you have more data and some hours during the night where you have less data. And the mean systolic blood pressure, if calculated as a straight mean, just averaging all of that data together, would be influenced more heavily by the density of data during the day. So a weighted mean is simply a way of balancing that to ensure that the influence is appropriately distributed, not just heavily influenced by an increased density of data during the daytime. In terms of change from baseline, generally when feasible to do so, it's best to do these in an open label design because the sample size is quite a bit smaller, as I mentioned before. And so there you're making a straight comparison from a 24-hour baseline ABPM session to a on therapy session. And you may have multiple on therapy sessions depending on your design. Placebo is recommended in the guidance now predominantly in the scenario where you have a much longer study, right? So if the duration is let's say 3 months, and you have a time point after 3 months of treatment, that's enough time for there to be some potential drift in the background that has nothing to do with drug effect. And you would need a placebo arm in order to detect that. So if you have a placebo arm, then you're correcting both for the individual patient baseline, but also correcting for change relative to placebo. Hope that helps.
Thank you so much, Jeff. How does this work? I mean, it sounds quite awful to have a blood pressure measurements go off twice per hour during nighttime. I mean, what are we looking at? Do we ever get that amount of data?
It's a good question, and we do have a great deal of experience, again, with the implementation of ambulatory blood pressure monitoring in this specific arena, which again is that increase, that safety assessment. The field of hypertension research has been using ABPM for many years. It actually initially started with inflation sequences of going off 4 times an hour during the day and 3 times at night. And then slowly, as the statistics and the comfort became more accustomed to it, the inflation sequences have changed. And as you can see in the draft guidance right now for this specific assessment, it's 2 times an hour around the 24-hour period. So study participant compliance is actually pretty good. The key to the compliance is really ensuring that both the clinical site is well trained in the use of the technology so that they in fact can then train the study participant in how to best live with the ABPM. In addition, the ABPM technologies are designed to quote unquote learn the individual's blood pressure. And as seen, you see that circadian rhythm with that dipping effect in the evening for a majority of the population. The blood pressure, the device learns the blood pressure and normally only goes 30 millimeters above the last recorded systolic reading. So I think compliance overall is quite good, even with the 2 times an hour around the clock.
Does the device alert the subject?
It will, it's a very good point. And what happens is that devices can be configured that during the daytime, it can inform the patient with a beep tone that it's about to take a reading, as Dr. Rudo shared that there is a question of motion artifacts. So with that notification, the individual can relax their arm and allow it to take the reading. Likewise, in the evening, those notifications can be turned off so that in fact it doesn't disturb the individual during the assessment period. Often what you find out is that the study participant, he or she becomes acclimated to the blood pressure assessment, and more likely it's the significant other or spouse who's like, what's that going off over the 24-hour during the nighttime period. So
thank you, Jeff. And Dr. Kleiman, what do you think about, let's have a drug for which you, in vital signs assessment or in clinical blood pressure assessment, can demonstrate that the drug actually decreases blood pressure. Is there then really a need for an ABPM session also for those compounds?
Well, I think it would depend on the effect size and just how confident you were that that was really an antihypertensive effect. And we know that there are people who have white coat hypertension. There are also people whose blood pressure is lower when they're in the office. Another problem with office blood pressures is you're taking blood pressure at one point in a day that may not be related clearly to the dosing schedule. So a patient may dose in the morning and have an office follow-up visit in the morning, at lunchtime, at the end of the day. So it may be very difficult to tell from just the sporadic blood pressure measurements in the office whether there's really a decrease or not. And I think you would need to demonstrate it clearly.
Well, absolutely. But I mean, in the early study, you have several PK draws, and you can have vital signs assessment paired with some of those PK draws. If you, with that approach, can demonstrate that you clearly have an expected blood pressure reduction, what is your take on that?
I think you would have to demonstrate that that is true after 4 to 8 weeks, because we know that many drugs have a different effect on blood pressure acutely and chronically.
Yeah, makes sense. Thank you so much. And we have, I'm very happy to say that we have received many more questions than we can address. And what I can promise you now, since we have reached the top of the hour, is that we will address those questions in written back to those who've raised the questions. And by that, I would like to thank you all for attending this webinar. I think this is a very interesting and emerging area. And again, please be aware that you can request, and we will also share the recordings from this webinar. So thank you so much to all attendees. Thank you to our panelists, and have a great rest of your day. Thank you. Take care.
Thank you.